All right, good morning, good afternoon, good evening, and good night. Hello, invertebrate enthusiasts. Welcome to Lecture 18. This is Monday. It's Monday afternoon. For those of you joining asynchronously, or for those of you who were here for lecture on Monday morning and are noticing things are a bit different, it's because my recording was nibbled at by gremlins and the audio didn't record. And thank you to those of you who let me know as soon as that was posted. That's super helpful to be able to do that because I wouldn't have noticed, it seems. So what we're going to do is I'm going to go through the lecture again, and I've got some screen grabs from the live session this morning that I'll show, and hopefully those will augment some of the questions and that we were kind of talking about. But here we go. We're talking about annelids. We've been in the lophophores, the Lophophorata, so the phoronids, the brachiopods, and the bryozoans. These kind of tentacle, coelom-filled tentacle, mainly sessile animals, some shelled, some living in zoocoelom. And for the next 3 lectures, nearly entirely, we might change over on Friday, but we're going to be talking about annelids, segmented worms, true worms. And that's kind of exciting to me, because it's the most diverse group by far that we've dealt with, and there's real kind of structural meanings to that. Their diversity is really caused by adaptations, morphological changes in the evolution of this group. So we'll talk about that. The first thing that I wanted to do is to unpack some thanks to those of you who participated in the Stop, Start, Continue survey on CourseLink. And I wanted to talk about that just a little bit before we go any further. One of the things that I've seen in that list of things that you would like me to stop doing is no more breakout rooms. Now, if you're attending asynchronously, this is like rather neither here nor there, but for lots of you who are attending synchronously, you've given me I've seen and heard and now will act on the feedback that things just aren't going well. And our hope is that in those breakout rooms, you're interacting and talking with each other, and then can jump over to the Google spreadsheet and share some of the discussion. But I mean, if discussions aren't happening, then yeah, that's it. So we're not going to do breakout rooms anymore. In place of that, whenever I had kind of imagined there to be active discussion, what we'll try and do is to foster that in the chat. And you'll see some of the results of that in some screen grabs that I've got below. The things that you'd like me to start doing. I heard from a couple of people that they're interested in links, in some kind of unified place where all of the links, the videos and the Twitter posts and the primary literature and all of that kind of appears. And that actually does already exist. And it exists in the chat. So the chat that's associated with each lecture essentially ends with me pasting all of the links that I've compiled that are contained within that PDF. There could still be some errors in that, some YouTube links that are no longer active. If you find that kind of a thing, send me the link and let me know, and I can update it. But I do my best to try and check those through to make sure that everything that I'm sharing with you is alive. And then the continue, there's lots of kind of thank you. There's lots of good comments about appreciating enthusiasm and energy, and that's great. And I appreciate yours as well. It helps. Let's help each other keep going. The pandemic will change this year, and we will begin to return to some of the after times. But in the meantime, we can be excited about things at a distance, but together. So let's today start talking about annelids. So you can see annelids, we're within the bilateria. We are within the spiralia, where we were last seen talking about, we talked about platyhelminth flatworms, parasitic flatworms, monogeneans, digenean trematodes, cestodes, or tapeworms. We talked about rotifers and chaetognaths, the nanthoferans, and we talked about bryozoans, and help me find them, brachiopods and phoronids. So we're making our way through, but now we've landed in the second most diverse group of the spiralia, and that's the annelids. These are segmented, bilaterally symmetrical, vermiform. We'll talk about that word in a section, but to break it up, verma means worm and form means shape. So it's a worm-shaped thing. There are protostomes, which has to do largely with their developmental stages. You've seen that in some of your readings in the Brusca text. We know them, or perhaps if you're a terrestrial biased like me, terrestrially biased person like me, when you think of worm, you think of an earthworm, which is kind of strange because where I'm sitting and the window I'm looking out in was only recently deglaciated, and so shouldn't have worms. And so all the worms here are recent immigrants that have been brought in by largely European colonizers in the past several hundred years. But in fact, it's kind of a weird perception in another way too, and that's because the vast majority of worm life, annelid life, is in the seas. It occurs as marine forms. So they're segmented. This is we're going to talk about segmentation and advantages that segmentation provides today. And when we don't see segments in annelid worms, it's because they've been secondarily lost, where the segments have been secondarily fused together, but they aren't there in the developmental processes that form them. They form by a teloblast growth. We'll talk about kind of a black, like a nice simple blueprint of what a worm would look like. They have a complete digestive tract, a closed circulatory segment, respiratory pigments, like a slurry of them that can include hemoglobin, chloroquine, hemathirin. We'll talk about more of that on Wednesday. They have metanephridia, paired metanephridia associated with segments, and well-developed nervous system. Secondarily, segmentally arranged chaetae, which help them with traction and movement, and they are generally tend to be comprised, as this whole clade is here, of annelids, mollusks, nemerteans, brachiopods, and pharanids by trochophore larvae, or larvae having that little mustache of chaetae. So the annelids, back in the 20th century, a long time ago when I took this class, we were taught about annelids as being comprised of three main groups. Polychaete worms, so the marine ones, the ornate kind of paddling parapods in the sea. Oligochaetes, or lacking parapods, earthworms, etc., in the terrestrial environment, and then parasitic forms like leeches. That has been reconfigured in some major ways. The first was the realization, phylogenetically or systematically, that the earthworms and leeches were, in fact, it wasn't possible to talk about them as two different groups, that they were only a monophyletic group when considered together. And then so we talked about Clitolata and Polychaetes for a little while. And then using molecular data, what we have found is that a lot of other recent evidence suggests that there's other taxa that we used to consider separate phyla back in the 20th century when I took this class, are in fact different kinds of worms, including sipunculids, cybogonids, pogonophrans. And so in light of these two major reconfigurations, we're going to talk about the fact that you may hear me use the word polychaete or leech, but these are common names now, and they don't have any kind of phylogenetic basis. Neither of them are monophyletic. And so what we're going to try and do is use the term Arantia and Sedentaria. And these are two clades within the annelids that are comprised and largely, as you'll see in the phylogeny that I'll show in a couple of minutes, predicted by their mode of life, whether they're active, the Arantians, errant, or they're sedentary and locked in place or not very great movers like the Sedentaria. But before we go much further, I want to take the chance to talk about this vermiform, this phrase vermiform, or worm-like. And I want to use this image that I nicked from Ross Piper in his blog post of 4 or 5 years ago, that is, a worm is not just a worm. In fact, a worm, if we use the word worm, phylogenetically, it's only monophyletic, essentially, if it refers to all of the dun phylogeny. Now, how can that possibly be? Well, the answer is actually here on the slide here. You can see all of these examples. These are all commonly called worms. So different xenoturbellins, different arthropods even, different cnidarians, different ctenophores. We've already talked about the chetognathe worms. None of these, except for the annelids, are true worms, but they all have species or taxa within them that are vermiform. And so 23 or 4 different phyla of the 34 odd phyla in the Dunn et al. Phylogeny, so roughly 2/3 could be called worms. So a worm, if we're going to call anything a worm, we have to call everything a worm. And I mentioned this morning, that made me think of a Churchill quote, where Winston Churchill, before the First World War, when he was having some difficulty in life, having people believe in him, he said, "I think we are all worms, but I do believe that I am a glowworm." Churchill showing you that he didn't have much of a problem with imposter syndrome. So here's a look at the cladistically what we're going to be talking about. So within the annelids, two major clades, Aurantia here in light green and the Sedentaria in blue. And their evolution is driven by adaptation to two different modes of life. In the Sedentaria, they have a clitellum, cocoons, and no appendages. Movement is not really the thing. Some small amounts of movement, as you can, so the earthworms are in here. They can move, but it's slow. And their dispersal range, their movement range is not large. The Aurantia have appendages, have parapods, and a nuchal organ, so they're much more active. They have a much better developed nervous system. They have a much better developed set of photoreceptors, even eyes, potentially image-forming eyes. And these parapods, these paddles, help them move around in the environment. So many species, leeches, earthworms, bristleworms, feather duster worms, pogonophrans or worms on deep sea vents, and then parapod containing polychaete-like things. But let's talk about first a little bit about the apomorphies for this group, and they are largely metameric segmentation. This is an enormous one, and it's hugely advantageous to the group and has been tied really elegantly, I think, to our understanding of why their diversity has exploded. So metameric segmentation is essentially the modularization on a longitudinal axis, so from head to tail, of the body. And so metameres are a septa dividing fluid-filled coelom, so segments that are replicated from along a longitudinal axis. They also have chaetae, and that are these bristles here. We'll talk about the chaetae and metameric segmentation in some more detail now. So what is metamerism? Well, this is the structural plan so that you divide the body on this longitudinal axis into a replicated series of units or segments that each, if you were to look at them in cross-section, contain elements of the chief system of organs. So excretory organs, they have a pair of nephridia, a pair of ganglia, a pair of pedal ganglia, a pair of appendages, a pair, okay? And so replication along the longitudinal axis. Now, if we zoom out to the whole view of the body, of the annelid body, we can break those, the body up into three different regions. The anterior end is here on the left-hand side of the screen, and we call that the prostomium. If this is a train of cars from left to right, this is the engine. This is the head. So there was cephalization, where photoreception, there's a pair of antennae up here, so lots of kind of modification of the body, of a bilaterian body plan to moving around and bumping into things. Now, segments are actually at the tail end, produced just in front of the pygidium. The pygidium is where the excretory system ends up. Your anus is there, if they have one, and so we've got the caboose at the far end, and just in front of that, and then in between, metameres, and these replicated segments of septa-divided, coelom-containing, replicated body segments. You can see pairs of parapods here, these little rounded nubs with the chaetae sticking out and paddling in the water. Now the growth region is just in front of, now this is mesoderm and ectoderm in the area just in front of the pygidium. So these metameres here at the tail end of the body, these are the youngest metameres. The oldest are up here by the head. And these are formed by teloblast cells that are, so these nearly totipotent cells that are forming segments and kind of budding off all along the way. And then the peristomium is this neck-like region between the peristomium, the head, and the metameres. So metameres, as we've said, contain coelomic spaces, pairs of nephridia, pairs of coelomoducts, which are ciliated excretory reproductive channels that we'll talk about in the coming days, pairs of ganglia arranged and connected along a ventral nerve cord, and pairs of appendages. Now, you might be sitting there furiously taking notes and thinking, well, we've seen a segmented thing. Just like, I think early last week, we saw the parasitic cestode, and that had segments, didn't it, Smith? Didn't it? Well, it did. Those were segments. Those are strobila that the strobila produced, or proglottids produced by strobilation in the parasitic cestode. So we've got this kind of locking on scolex here and all these bags of eggs. And this is one of the key differences. And I'll show you an arrangement of them in a second. But these cestode proglottids form anteriorly. Remember, the oldest are down here and the youngest are up here. And the annelid segments form in the growth zone in front of the pygidium. So they form from the posterior and grow towards the anterior. They're developmentally very different. One forms from the ectoderm, one forms from the mesoderm and the ectoderm. And these are functionally very different as well, and ought not to be confused. So within the metameric segmentations, they're derived from the mesoderm, and they comprise, as we've talked about, these replicated segments. And so all these key elements of the architecture of the animal are replicated in pairs along and within each segment. And that's not at all what we see with the kind of proglottids segmentation within the cestodes. They're derived from a different developmental tissue, and that's only confined to the ectoderm. And so they're essentially bags of eggs that appear to be segmented, but are in fact not. Now, this is one of the times in this secondary recording of for the asynchronous attendees, I asked people to consider what some of the advantages of metameric segmentation might be. If you want to play along in this kind of a way, what I would suggest is pause the video and maybe jot out a couple of reasons why you think, what advantages to an individual organism living in the sea might be to be metamerically segmented, and what advantages that might provide ecologically, and if those would then extend through time evolutionarily. So I'll let you pause. And I'll continue. And I'll show you, now that you've been through that bit of self-reflection, you can see what your compatriots this morning came up with. And it was a whole bunch of, if I can capture most of what I saw in this morning's chat, it was about redundancy. Redundancy of organs and organ system, leading to an increased survivability due to damage. So if there were bits that were nibbled off, the organism could continue to function and continue to grow. So redundancy, increased survival, leading to, for predation, increased efficiency, with this person talking about the surface area to volume ratio. And we then reconvened, and I talked about some of the ways that I categorize advantages of metameric segmentation. That you can build a very large organism from a very simple plan, just as you can build very large structure from individual kind of the same sorts of Lego blocks. These are essentially Lego blocks that have building ends, a building end here and a bit of a head and a super attachment at the interior end. You can effectively service major systems because you don't have a pair of nephridia that is trying to drain all the metabolic waste from a 3-meter long worm. You have every couple of segments, maybe every couple of centimeters, you have a pair of nephridia that are trying to drain and deal with excretion and osmoregulation. So it's much more efficient. It also allows, as your compatriots talked about, loss of parts is not fatal, so redundancy. It also enables flexibility and complex movements because you're kind of reticulated. And this is a really important one at the tail end, that it allows opportunities for specialization within those segments, so that some segments can be specialized for some functions. And so that we see, even though there are structures, and so we see this feature or this name applied to annelids in terms of being metamerically segmented, but also serially homologous. So these are the same structures that, although they appear different, although the parapods may on this part of the body just be about moving, and on this part of the body back here be about respiration. So although they're different, they have the same segmental, embryonic, and evolutionary origin. So that's serial homology. And it refers to segments and appendages. Now, whether you've run into this or not, you know it. And you know it, I know, because probably you could all sit down and draw me an ant. And you draw 3 circles. You draw a head, and you draw a thorax or a body, and an abdomen. Now, those, in fact, are grouped segments in the arthropods, which are also metamerically, so are also segmented. But the metameres in arthropods are grouped into what's called tagmata. So the tagmata are structurally differentiated groups of segments, the head, the thorax, and the abdomen. And so this tagmatization is kind of an extreme end of serial homology. So you can have serially homologous metameric segmentation. There is nobody here in the chat, so we'll just not vérifier le champ. We'll keep talking about the apomorphies that include these chaetae. So these are cuticular structures that are produced by epidermal cells that are chitin, in this case beta chitin, and they're essentially small bundles together of sclerotinized chitin that stick out of the body and out of the parapods if they are there and allow traction, and in some cases other tasks, but principally traction. Now, I mentioned here, and we'll come back to this when we get to the arthropods in a few weeks, that there's two different kinds of chitin. These are on the left-hand side, we have beta chitin that makes, you can see a cross-section of them here. So bundles of sclerotinized chitin that are kind of bundled together where the molecules of chitin are aligned in parallel lengths, and it's tough and flexible, and that's the chaetae. And then the alpha chitin, which is hard and stiff and more like a shell, that's this pulpy cavity, and the chaetae is then derived in arthropods through a slightly different alignment of this molecule. We'll talk about some of the difference for alpha chitin, consequences of that, when we get to the arthropods later on. But there they are. So an enormous amount of diversity here. And so we've got Arantians, Polychaetes, or sedentarians, Annelids, and leeches, and then other groups that historically would have been considered their own individual phyla, but we now know because of molecular evidence that their absence of some classic annelid traits like segmentation is secondarily derived. Now let's talk about some of these other adaptations or apomorphies that characterize annelids in general. So serial homology serial metameric segmentation, chaetae. Now also parapods. These are these paddles that are sticking out here and many times paired on each one of the metameres. And they're muscular. They're derived from the vagination of the body wall, but they're muscular, and they're generally used for locomotion, but also can be sensory or respiratory. Each one of them contains a little pedal ganglia, which we'll talk about in a second. But we'll begin talking about the fact that in the cephalization of this group, with the concentration of the nervous system, replication and ganglia along each segment, but then a concentration up at the anterior end, and lots of different receptive capacities. So there's your parapodium again, but here, nestled in kind of right in your neck, the neck of that, just behind the prostomium, is the nuchal organ. And the nuchal organ can be involved in chemoreception. It can be ciliated or pitted or a groove. It can be involved in light detection, and a lot of these are involved in detecting food or avoiding predation. They can be raised up from the body. Here's one from your figure 14.6 in your text, where the nuchal organ has been raised up and isn't kind of tucked into your shoulders here, but is raised out to facilitate the collection of whatever chemicals are in the water that it's trying to sense. Now, that concentration is associated with a whole bunch of different, particularly in the marine environment, a dorsal, an interior and dorsally located brain, where a pair of ventrally aligned nerve cords fall back from, and then bud off into parapodial ganglia along each metameric segmentation, and can also include eyes. Maybe even image-forming eyes. There's one marine polychaete or Arantian shown here, but this eye is basically, you can see all the same kind of components that we have, where there's a lens and an iris and retinal cells and sensory cells that lead to an optic nerve. So clearly, these are visually aware organisms. We're not entirely sure yet if they're image-forming eyes, but they are certainly capable of detecting very subtle changes in the light environment. Now, those subtle changes are also then backed up in an ecological and an evolutionary sense with giant axons that we see again, particularly within the marine Aurantians, or formerly called Polychaetes, where they have these giant axons. Here's one particular example, the Myxicola infundibulum, where this little circle down here, this is a ventral, the largest nerve fiber known in the animal kingdom. It can reach a diameter of about 1.5 millimeters, which is as large as the body size of some entire body size, larger than the body size of some other invertebrates. And they have a large diameter, low resistance, so rapid conductance, and their conduction velocities can meet or exceed our own. And we believe this has to do clearly with escape responses. So there's these pedal ganglia now. You can see in this particular, in figure 14.15, the little bulb there of the nervous system branching out, in this case, into the first parapodial segment, so the oldest parapodial segment, so the most anterior. And then at the tail end, we have one apomorphy for the Aurantians, the Polychaetes, this pagidal cirri, or these sensory appendages at the at the posterior end near the anus. Now, for the Aurantians, we have, or the three different, they can be burrowers, they can be crawlers, they can be swimmers. And this has to do with the fact that much of this movement is, even though there's no hard body, really hard, there's some, the Chaetae is kind of hard, but we're not dealing with like a chitinous exoskeleton like you can, and so levers like you can see, or we will see later on within the arthropods, but they're essentially soft bodies. And so the same kind of hydrostatic skeleton that we've seen back to the Cnidarian is still roughly what's functioning here. And one of the major differences is that we've got more musculature systems, more systems of muscles that are firing on this closed hydrostat or coelom. So we've got not just circular and longitudinal muscles, but we've got oblique and parapodial muscles that are capable not just of squeezing it and allowing it to inch forward, but in changing orientations and in moving the paddles in waves and in segmented and coordinated waves. And that's possible because of these oblique and parapodial muscles, but also because of the pedal ganglia coordinated through this long ventral nerve cord that allows the coordinated firing of waves. Now, this is a bit of a callback to what we saw with the ciliofferents. Remember the metachronal waves proceeding across the cilia as wind across a wheat field that one of the authors of our previous text waxed lyrically about? Well, this is another case where we're seeing coordinated firing of these parapodia. And we'll see more, some good video of that in Wednesday's lecture. Now, the cuticle that supports this body, the fibrous, the fibrous cuticle that supports this body is aligned, which reinforces and prevents bulging when the hydrostat is fired upon by these muscles, isn't laid at right angles entirely. It's right at crossed helical orientation, and that's because if they were just orthogonal, kind of with right angles left, like an X and Y axis on a graph, when they're bent, you would see kinking, and that could pinch and break, or it could impede any of the modular systems of excretion or nervous system or reproduction that were kind of arranged longitudinally across the length of the animal. But if we kind of take that and rotate it slightly, when they're kinked, when they bend, they don't kink, and they have an elegant kind of a sausaging effect instead of a what would that be? I don't know. Yeah, a clothespin effect. Now, what kind of motions are we talking about? I think still some of you may be thinking about the kind of the inchworming of an earthworm. But let's have a look at some of the beautiful ways that polychaetes, that Aurantians can actually move in the marine environment.
This, though, has to be the most extraordinary animal design of all. It's a polychaete worm, and normally you would expect the long pulsating body to be stuck firmly in the sediment. This worm, alone in its group, swims in the open water. Propelling itself with its yellow frill, it moves about and so finds new sources of food, or maybe succeeds in escaping from a predator.
Amazing. I think I'm calmed slightly by listening to Attenborough like that. But that's from the first Blue Planet. There's a link there in the bottom if you want to go and check out the entire episode about the deep sea. So swimmers and burrowers, remember we talked about. So many polychaetes, many Arantians, sedentary ones, secrete tubes. And they build them from a bunch of different materials. They can be self-generated fibrous proteins, mucus, calcium carbonate, and in all cases, they end up mixing those materials with substrate to harden and to reinforce the tube within which they are growing. Now, the deposit feeders, these sedentary polychaete ones, they can be indirect or direct. They can actually ingest sediment and then excrete that which does not contain food. Or they can be indirect, like these amphiprites that collect organics via these tentacles and kind of move them into the mouth. We have other ways if you're living in a food, though. If you're living in a tube, doesn't mean that you are necessarily an indirect a sedentary. So I was going to say it doesn't necessarily mean indirect. It is still indirect what you're going to see, but it is certainly more active. So don't think of these as just passive, because there's some of these tube restricted polychaetes that are predatory, quite efficient predators. Some of you maybe know where we're going with this, but they can evert a pharynx that can be used as a grasping tube or containing very powerful snapping jaws. And that pharynx is then everted through the contraction of body wall muscles, largely circular, and the subsequent increase in coelomic pressure that shoots that head out. And so these, you can see jaws and a poison gland. And so thinking about worms that this morning reminded a lot of students of the Sarlacc worm from The Mandalorian or from Star Wars, or the worm that tried to capture the Millennium Falcon in The Empire Strikes Back, or indeed maybe of the as a sandworm if you're thinking about Dune or Tremors. So lots of pop culture references came up in the chat this morning. So here's a look at that feather duster or cebalid worm, where they construct these membranous tubes and then capture particles in a mucus web. So that's from a paper that's nearly as old as I am. But we also have this one to deal with. So not the tiny little octopoid, but that hidden sand striker worm that consumed it. So these sand striker worms take the idea of a sit and wait predator and just make it elegant and beautiful. So we're going to take a second and watch a Smithsonian video about these sand striker worms here.
With multiple mouth parts displayed, bone-hard grappling hooks poised to pierce and immobilize prey. But the real nightmare remains unseen. Coiled, pent-up aggression as long as an arm. And some specimens grow to twice the length of a man.
By which they mean 2 to 3 weeks.
As night falls, many sand strikers emerge to feast. It's a bad neighborhood to hang out in.
If you're a fish.
But fish are fast, and the sand strikers are feeding blind. They don't have eyes or a brain. They react only if one of their antennae is triggered or if a shadow lingers.
There's a great view of how the mandibles are invertible.
Patience.
This is them reappearing. Oh, that's gorgeous. Look at that.
Gotcha.
Yep. Good night, little fishy. So, the sand striker worm, or Eunice Aphrodite's, this lives in mucus-lined burrows in rocks or sediments, and these jaws are armed with sharp teeth. They have Photoreceptors, so 2 eyes. The video said no eyes, but there are 2 photoreceptors on the head. And 5 separate antennae that exhibit a worm-like movement that presumably help lure prey. But then as soon as they're tickled, bam, the jaws fire. So during the day, they are buried in the loose seabed waiting for something to stimulate one of those 5 antennae to slam the jaws shut. And at night, they change their strategy slightly and avert themselves to reach out into the water column a little bit. So imagine a very fast way to end. Now, one of the neat things about these worms and about our province, where we don't see them anymore, is that we would have, 450 to 500 million years ago, Ordovician to Silurian to Devonian, so up to 350 million years ago. The tropical seabed that characterized different parts of Ontario. Near Moosonee, there was a geological survey in 1994 that Derek K. Armstrong ran, where he found some fossil evidence for what he called Webster O'Brien Armstrongi, which is a many meter long giant sand striker worm. Could have been 1 to 2 meters long, and they're estimating this based on the size of the jaws that they found. Now, a neat thing, since we've talked about naming, the species part of that epithet refers to the geologist Derek Armstrong. Webstero prion. Prion means saw, and the Webster refers, because the taxonomists were some metalheads, it refers or honors, is an honorific for Alec Webster, who is a bass player for the death metal band Cannibal Corpse, a giant of a bass player, for which they named their giant of a sand striker worm. Now, one of the neat things, so there's 2 neat things here, many neat things, of which 2 of them are, these remains were housed in the Department of Natural History at the Royal Ontario Museum. So we have some ownership of these things. They're from Ontario, from near Moosonee. But also, it's neat to me that, in fact, the size is not really unique. In fact, there's contemporary examples of multiple meter long sand striker worms. Here's a paper that a lot of people haven't run into, and it seems to me, in that literature, from a small journal in Japan, where a live sand striker was found just off of a dock in a fishing village that was 3 meters long. And as the authors talked about this data, they compiled a bit of a survey of the literature and found over the last couple of 100 years numerous examples of sand striker worms that were longer than 1 meter up to 3 meters long. So these can be giants. Modular, metamerically segmented giants. Now, here's something for you to think about. Pardon me and my cough. Think about what these fish are doing. That first fish that you saw, the Siphinus, it's been blowing at what is a hidden sand striker worm right here. And she's alerting the rest of her school, the conspecifics in her school, maybe relatives, to the presence of that worm. And what happens, what these authors wrote about in 2019, was essentially a mobbing behavior, that enough of them, once one of the fish had discovered the sand striker worm and weren't killed by it, they began to blow at it, kind of ridicule it, if you want, and then others would join in, and that would cause the worm to retract. And then that essentially secured foraging territory for the fish. So what we did this morning, and what again you can do now asynchronous if you want, is pause me after I explain. But what I asked people to do was to explore what this graph would look like. So if there are social benefits to a conspecific fish spitting and disturbing the sand striker worm, what would you expect the relationship to look at when the x-axis here is the fish? The first category is fish that finds the worm. Then we have the first participant that joins the spitting fish. So first spitting fish, first joiner of the spitting fish, and then all subsequent fish that join in and spit at the sand striker worm. And then the y-axis is how many water jets would you that are shot by each one of those categories. So pause me and take a minute to maybe think about whether you would expect to see no relationship, an increase, or a decrease. I'll do And I'll share with you what the students this morning thought. And there was contrasting hypotheses. Most people tended to think that it ought to go up. Some people thought that it should go down. And in fact, what the authors found was down. And if you think about it, if there's conspecific benefits, social benefits to doing this, they should accrue in the fact that if you happen to be the one that finds it, it takes you some work to show it, to demonstrate to your conspecifics that you found it, you're spitting, and to get the attention of the predatory worm. But every subsequent participant that's added should have to devote less and less time to doing that spitting. They're able to take advantage of the fact. So there is a shared benefit to the school if one of them invests. Whoever finds it first invests quite a bit. And this was a neat study that was published, I think, in one of the Nature journals in 2019. And one of the things that I always thought was neat about the study was the fact that the authors excluded cases where the spitting fish was actually eaten by the worm. And so I'd like to see if you could go down to the Philippines and replicate this study. I'd like to see where cases with predation, which are not shown in their data, are they evenly distributed across these categories, or are most of them in this risky first fish that finds the worm first? Now, the last thing that I'm going to talk about before we break off this second recording is the fact that some of you may have been looking at the sand striker worm or watching those videos and go, I know that worm. Why is he? I know it by another name. And the other name that you may have heard the sand striker called is the bobbit worm. And that's problematic. It's a common name, and it's a problematic one because it is essentially an honorific for an abuser, and a rapist. And there is no reason that I can see that we need to teach each other to use names that, especially common names, that have no real kind of traction in the scientific community, I would argue, particularly amongst the invertebrates, where the vast majority of species don't have common names. There's no reason to apply a common name that essentially is an honorific for a rapist and an abuser. So if you watch these videos, you might hear the word bobbit, and what I would encourage you to do is to watch a documentary about Lorena Gallo that is on Amazon, and think good thoughts about her, and don't give any traction at all to the abusive rapist that she lived with, and call this one the sand striker. It deserves better than being associated with that name. So there's some literature that you can read about if you're interested in some of this. And that's it. That's all she wrote. And we will see you on Wednesday, where we're going to keep talking about annelids. So take care of yourself, take care of others, be healthy, and we will see you on Wednesday. Ta-ta.